Yeah. So as one of your friends said, Rhizopus. So the next topic, what I'm going to take is the common bread mold, that is Rhizopus. And if you see this Rhizopus, it is a genus of saprophytic and parasitic fungi. So it can be, it is generally a saprophytic fungi. I think you know, like it commonly grows on the bread mold, bread, and it is considered as a laboratory contaminant, but sometimes it can also be parasitic. And if you look at the ecological significance or the persistence nature of this fungi, they are found on moist or damp places. So that's why generally it is seen growing on the bread, because bread is a substrate that is rich in carbohydrates. And they are also found on organic substances like vegetables, fruits, bread, jellies, etc. And the vegetative structure, they have cenocytic hypha. Hypha meaning, like you know, it is the mycelial network, and they don't carry any cross wall. And they are also known to produce different types of chemicals and alcoholic products. So it also has very good industrial applications. And some species of Rhizopus, they can cause plant diseases and are also known to infect the human beings. For example, the disease mucormycosis, it is generally caused by the Rhizopus species. So it is an opportunistic fungal infection. So this is a small introduction on Rhizopus. So Rhizopus, they are commonly referred to as the bread mold. So generally they are parasitic in nature, but sometimes they are saprophytic in nature, but sometimes they are also parasitic. And now if you look at the systematic position of this Rhizopus family, if you see, they come under the kingdom Mycota, the division Eumycota, and subdivision Zygomycotina, and classes Mucormycota, and order Mucorales, and family Mucoraceae, and the genus is Rhizopus. Okay, so this is the general systematic position of this genus. And if you look at the important species of Rhizopus, you have the first very famous species, that is Rhizopus stolonifer. It is also known as the common black bread mold. And it is also commercially used in the production of different types of acids. And it can also cause fruit rot disease, which means that it is also considered as a plant pathogen. And if you look at Rhizopus oryzae, it has a very good industrial application wherein it is used in the production of different types of alcoholic beverages. Then you have Rhizopus microsporus, again, which is also used in the production of different types of fermented products. So these are the different genera, different species of Rhizopus. And if you look at the structure, so I think you would have studied this in school. And if you see, this is the basic structure of Rhizopus. So here, if you see, it has a globose-shaped cell that is called as the sporangium. And this sporangium, like it contains the spores, which are called as the sporangiospores. And if you look at the hypha, they don't carry any septa. So hence the mycelium, it is generally considered as cenocytic. Okay. And then if you see this, they have different types of hypha, which is called as, this region, you call it as the stolon. Then you have the root-like structure that are called as the rhizoids. And then you have a stalk-like structure, which is called as the sporangiophores. So the stolon, it is generally considered as the internodal region, which means that it will go and touch the substrate, and then again from this another Rhizopus will grow. So this region, you call it as the stolon. And then rhizoids, they are formed where the stolon will touch the substrata. So when it touches the substrata, that time it will form the root-like structure that is called as the rhizoids. And these rhizoids, they are helpful in the absorption of food. And then they also have the aerial stalk-like structure, which is called as the sporangiophore. Okay. And then these end in a branch, which is called as the columella. And then they have the sporangiospores, which is held in a sac-like structure that is called as the sporangium. And if you look at the cell wall of this Rhizopus, it is generally made up of chitin. Okay. And it is also made up of other types of organelles like mitochondria, endoplasmic reticulum, Golgi body, gas vacuoles, and oil droplets, because it is basically eukaryotic in nature. And if you look at the reproduction of this fungi, they can reproduce either by vegetative method, asexual method, as well as sexual method. And if you see, this is the thread-like structure, which you can see it under the high power objective of the microscope. So they don't carry any cross wall. Okay, they have cenocytic hypha, and then they also carry the nucleus. You can see this round-like structure, no? It is called as the nucleus, and the nucleus is basically haploid. And this is the basic structure of the Rhizopus. You can see the internodal region that is generally known as the stolon. Okay, and then this is the columella, Then this is a stalk-like structure that is called as the apophysis. Then these are the this is a sac-like structure called as sporangium, and inside that you have the spores that are called as the sporangiospores, and these are the root-like structures that is called as the rhizoid. So what is the use of this rhizoid? It helps in the absorption of nutrients. And if you look at the life cycle of Rhizopus, as I told you, it mainly reproduces by 3 different methods. One is the vegetative method, then you have the sexual method, and then you have the asexual method. So vegetative method, it is very, very simple. So it is by fragmentation, and each of the fragment of a stolon will develop separately, making a complete mycelia. So it will just break and it will give rise to a new mycelium. And asexual reproduction, it is by the formation of 2 types of spores. So first one was the sporangiospores, and the other one is known as the chlamydospores. So what is this sporangiospores? Sporangiospores, they are formed terminally in the sporangia of the aerial mycelium that are called as the sporangiophore. So 4, it is the stalk-like structure. So on that stalk, you will have the spore formation that is called as the sporangiospores. And these sporangiospores, they will be formed only under favorable conditions. So they are not formed under unfavorable condition. They are generally formed under favorable conditions. And if you look at the how does it form? The sporangiophore will develop from the upper side, okay, at the rhizoidal region, and the apical part will swell up, giving rise to the sporangium, okay, and the cytoplasm as well as the nuclei, they will move apically. Apically meaning they'll move in the upper direction, and then there will be a central columella region formed with fewer nuclei and more vacuoles, and these sporangiospores will develop inside this swollen structure that is called as the sporangia. Okay, and these sporangiospores, they are multinucleated and they are generally non-motile. And these spores will, the sporangium will rupture after maturation, and the sporangiospores will come out as a powdery mass. So that is what you can see on the bread. Okay, it comes as a powdery mass. And after suitable favorable conditions written, they develop into a vegetative mycelia. And similarly, chlamydospores, they are not formed under favorable conditions, but then they are formed under unfavorable condition. Okay, so in this, an intercalary segment of mycelia will develop due to the formation of septa and the accumulation of protoplasm. Okay, and it is thick-walled and it will detach from the mycelia once it dries. Okay, so here there will be septa formation and there will be accumulation of the protoplasm, and it becomes a thick-walled structure. Okay, and then it will detach from the mycelia after it dries. And these chlamydospores will remain dormant until the favorable conditions return, and then they will germinate to give out a new mycelia. So these are the 2 different types of asexual spores that are formed by the Rhizopus. Okay, so the Rhizopus species, it can divide by 3 methodology. One is fragmentation and asexual method, where it forms the sporangiospores as well as the chlamydospores. And if you look at the sexual reproduction, I think I've taught you already. So they are heterothallic in nature. Heterothallic meaning one will act as the plus trait and the other one will act as the minus trait. So what will happen? The compatible hypha will come together, okay, and then there will be an outgrowth taking place, which is called as the progametangia. And the progametangia will again divide by a septa that is called as the gametangia, and these gametangia will conjugate to give out a multinucleated structure. And there will be a process of plasmogamy followed by karyogamy, wherein a diploid zygote is formed, and these zygotes will carry the zygospores, and the zygospores, they will enlarge and form thick-walled and resistant to adverse environmental condition. And on return of the favorable environmental conditions, the zygospores will start developing into, they will germinate, giving rise to the zygospores, and then meiosis will occur and haploid meiospores are formed, and they come out after the rupture of the sporangium wall and they develop into a new mycelia. So this type of reproduction, you call it as sexual reproduction, and it is a classic example of heterothallism. So this is what I've given you in diagram. So you can see the sexual reproduction. There is a plus strain, there is a minus strain, right? And then there will be a swelling happening from the plus and minus strain. This is more or less similar to the process of conjugation, and the nuclei from both hypha, okay, it will start moving together towards the center. Okay, you can see here, okay. So it forms the progametangia. Then later on, it matures to give rise to the gametangia. Then these 2 gametangia will fuse together to give the zygote nuclei, and these zygospores are formed by the process of meiosis. And then if you see the zygospore, it will form around these nuclei, and the zygospore will germinate when the favorable condition results. Okay, and then the hypha will be later on developed from the zygospore, okay, with the sporangium at the tip, and the sporangium will open, releasing many haploid spores, which will later on develop into new individuals. And this is again, I have put it as a life cycle where you can see the mating type, the plus type and minus type. There is formation of gametangia. You can see the central one is red in color and the other one is green in color. They are fused together to give rise to the gametangia with haploid nuclei. Then there is an young zygosporangium form. Then there will be a zygosporangia. You can see this is a microscopic image, okay, where you can see the formation of zygosporangia. Okay, and then again by the process of karyogamy, there is formation of diploid nuclei, 2 in zygote. Then there is meiosis, and then there will be liberation of the spores. And upon return of the favorable condition, the spores will germinate, giving rise to the vegetative mycelium, and there will be formation of asexual reproduction. Okay, and thereby the rhizopus is formed completely. So this is the entire